Welcome back to The Explainer. Today, we're doing something pretty amazing. We're going to shrink right down to the microscopic level to explore the absolute cutting edge of avian physiology. We want to uncover exactly how these incredible creatures manage to survive, and honestly thrive, in some of the most extreme, unpredictable environments on our rapidly changing planet. Okay, let's dive into this, because right off the bat, we're faced with a really visceral biological paradox. Just think about it for a second. How do birds, and we're talking everything from domestic chickens in a high-stress poultry facility, all the way to penguins out there braving freezing, high-altitude climates, how do they adapt so incredibly fast to climate shifts, devastating diseases, and, spreading urbanization? Like, how are their bodies actually adjusting in real time? Well, for the longest time, we tried to answer that question using a pretty flawed approach. The old way, that 20th century model of biology, was completely siloed off. You literally have ecologists in one building, geneticists in another, and physiologists somewhere else entirely, all studying the exact same bird, but totally in isolation. But the new way, the actual path to understanding adaptation, is what we call multiomics integration. We have to view biology for what it is, a complete, fully interconnected system. To really grasp this, I want you to hold on to a specific mental model for the rest of this explainer. Think of it as a biological symphony. Imagine the outside environment, say a sudden heat wave or a brand new pathogen, as a really demanding audience. Now the bird's DNA, the genome, that's your static sheet music. The transcriptome, that's the conductor, actively reading that music and deciding exactly what needs to be played right in that moment. And the proteome, well, those are the physical instruments actually playing the notes and doing all the heavy cellular labor. So let's look at Act 2, Part 1, Genomics, the core blueprint. Now this is our sheet music, and to truly understand it, we first need to shatter a pretty common biological misconception tied to a massive number, 60,000. See, across all eukaryotic life, genome sizes vary immensely by over 60,000-fold. For decades, scientists leaned on this assumption called the C-value paradox. It essentially guessed that a larger genome automatically meant a more complex organism. But as you'll see when we look at birds, that rule gets thrown right out the window. Now, what's really interesting about what we're looking at here is that, unlike mammals or plants with these huge, sprawling genomes, the avian karyotype reveals a surprisingly compact and stable genome. Why is that? It's because they have a brilliantly low occurrence of transposable elements, that quote-unquote junk DNA that puffs up the genomes of other species. This sleek, ultra-compact sheet music directly drives their famously high metabolic rates, which is absolutely crucial for meeting the extreme energy demands of flight and, of course, rapid adaptation. Moving on to Act 2, Part 2, Transcriptomics, reading the code. But, as we know, sheet music can't play itself, right? This is exactly where transcriptomics steps onto the podium. Simply put, transcriptomics is the large-scale analysis of mRNA levels. If the genome is that static sheet music that never ever changes, the transcriptome is the total opposite. It fluctuates wildly. It acts as our conductor, taking in internal and external environmental cues in real time and orchestrating a super dynamic response by turning specific genes up, down, on, or completely off. I mean, these gene expression heat maps and clusters might look a bit like abstract art, but this data is literally the conductor waving their baton. When an avian tissue, say a chicken's breast muscle or a penguin's liver, experiences environmental stress, this real-time shift tells us exactly which mRNA instructions are being frantically sent out to the cells. It's happening right before our eyes. Which brings us to Act 2, Part 3, Proteomics, the functional biological machines. These are the actual instruments playing the music. And so the crucial point here is a massive, totally counterintuitive truth that honestly shocks a lot of people. There is little to no correlation between mRNA and the levels of its corresponding protein. Just let that sink in for a second. Just because the conductor, the transcriptome, sends out an mRNA instruction, it doesn't automatically guarantee that the protein instrument is actually built or even active. Crazy, right? To really see what's happening on the ground, we absolutely have to measure the proteins directly. And while sure, the biochemistry itself is super complex, the actual workflow is incredibly sleek. It really breaks down into an approachable 1-2-3 sequence. First up, you've got complex protein extraction from the tissue. Second, breaking those proteins down into measurable peptides. And third, running the whole shebang through powerful mass spectrometry analysis. And this brilliantly illustrates exactly how proteomics takes what is essentially a chaotic, deeply tangled web of millions of interacting proteins inside a bird's cell, and using that mass spectrometry, completely untangles it into readable, actionable data points. We can literally map out the functional networks responsible for things like disease resistance or muscle development. It's a real game changer.
Now for Act 3, the revelation and action, synthesizing the Multiomics Symphony.
This is where we visually harmonize the static sheet music, the dynamic conductor, and the hard-working instruments into one complete, beautiful system. Because, ultimately, you might be wondering, why does all this microscopic mapping actually matter? Well, it matters because true biological understanding requires harmonizing all three of these pillars, genomics, transcriptomics, and proteomics, to solve immense, real-world problems. By understanding the whole system, we can accurately diagnose avian stress, combat devastating agricultural diseases, and really build climate resilience for both wild conservation efforts and global poultry production. For the veterinary professionals, researchers, and PhDs tuning in with us today, there are 3 clear, actionable takeaways from this integrated science. First, we've got to move past just treating isolated symptoms and fully embrace integrated omic diagnoses. Second, we can now actively leverage those compact genomic markers we talked about to selectively breed birds for true climate and disease resilience. And third, by utilizing proteomics, we can target early disease detection right at the cellular level, long before any outward clinical signs ever appear in the flock.
So to strip away all the heavy jargon for a second, just think of a bird like a smart house. The genome is the electrical wiring built into the walls. The transcriptome, that's the smart thermostat on the wall, making real-time decisions based on the weather outside. And the proteome represents the actual heater and air conditioning units turning on and doing the heavy lifting. If you want to truly understand and fix the house, you can't just stare at the thermostat. You literally need to understand all 3.
Which leaves us with this final thought. If this incredible multi-omics approach can decode the rapid survival strategies of birds, how will mastering this biological symphony across all species prepare us to navigate the catastrophic environmental challenges of tomorrow? The microscopic tools are finally in our hands. Now, we just have to learn how to listen to the music. Thanks for joining me on this explainer, and I'll catch you next time.